Hello guys, welcoming you all. Today we are going to see about material characterization. Unit 2. Spectroscopic technique. Raman spectroscopy. Raman spectroscopy is a non-destructive chemical analysis technique which provides detailed information about chemical structure, phase and polymorphy, crystallinity and molecular interaction. It is based upon the interaction of light with the chemical bonds within a material. Raman is a light scattering technique whereby a molecule scatters incident light from a high intensity laser light source. Most of the scattered light is at same wavelength as the laser source and does not provide useful information. This is called Rayleigh scatter. However, a small amount of light is scattered at different wavelengths which depends on the chemical structure of the analyte. This is called Raman scatter. A Raman spectrum features a number of peaks showing the intensity and wavelength position of Raman scattered light. Each peak corresponds to a specific molecular bond vibration, including individual bonds such as carbon to carbon, carbon double bond carbon, nitrogen to oxygen, carbon to hydrogen, etc., and group of bonds such as benzene ring, breathing mode, polymer, chain vibration, lattice modes, etc. Information provided by Raman spectroscopy. Raman spectroscopy probes the chemical structure of a material and provides information about chemical structure and identity, phase and polymorphism, intrinsic strain, contamination and impurity. Typically, a Raman spectrum is a distinct chemical fingerprint for a particular molecule or materials and can be used to very quickly identify the material or distinguish it from others. Raman spectral libraries are often used for identification of a material based on its Raman spectrum. Libraries containing thousands of spectra are rapidly searched to find a match with the spectrum of the analyte. In the combination with mapping Raman system, it is possible to generate images based on the sample's Raman spectrum. These images show distribution of individual chemical components, polymorphs and phases, and variation in crystallinity. Raman spectroscopy is both qualitative and quantitative. The general spectrum profile, peak position and relative peak intensity, provides a unique chemical fingerprint which can be used to identify a material and distinguish it from others. Often, this actual spectrum is quite complex, so comprehensive Raman spectral libraries can be searched to find a match and thus provide a chemical identification. The intensity of a spectrum is directly proportional to concentration. Typically, a calibration procedure will be used to determine the relationship between peak intensity and concentration, and then routine measurements can be made to analyze for concentration. With mixtures, relative peak intensities provide information about the relative concentration of the components, while absolute peak intensities can be used for absolute concentration information. Raman is used for microscopic analysis. Raman spectroscopy can be used for microscopic analysis with a spatial resolution in the order of 0.5 to 1 micrometer. Such analysis is possible using a Raman microscope. A Raman microscope couples a Raman spectrometer to a standard optical microscope, allowing high magnification visualization of a sample and Raman analysis with a microscopic laser spot. Raman microanalysis is easy, simple. Place the sample under the microscope, focus and make a measurement. A true confocal Raman microscope can be used for the analysis of micron sized particles or volumes. It can be even be used for analysis of different layers in a multi-layered sample. Example: polymer coating and of contaminants and features beneath the surface of a transparent sample. Motorized mapping stages allow Raman spectral images to be generated, which contain many thousands of Raman spectra acquired from different positions on the sample. False color image can be created based on the Raman spectrum. This shows the distribution of individual chemical components and variation in other effects such as phase, polymorphism, stress strain, and crystallinity. Thanking you.